This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1087, recorded on February 9, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and everybody else. The last couple of shows, I looked out the window and it was absolutely grotty out. It was just miserable with rain and fog and cold and bone-chilling. Today feels like the first day of spring. The sun is shining. The temperature is over 50 degrees, which is an anathema. This is absolutely unwelcomed at this time of the year. The trees, we have crocuses in our garden outside of our apartment that are now starting to throw shoots up out of the ground.
Dixon, an anathema is something you don't like. That doesn't make sense.
I don't like this at all.
Oh, okay.
I do not like this. This is like, what the hell happened to winter? And the answer is...
It'll be back. Don't worry. It'll be back.
Well,
here in the city, Dixon, it's 11 Celsius and sunny. It's a very, very nice day.
It is a very nice day.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We got 75 degrees, and the cloud indicator Tinkerbell is absent. So you can tell it's cloudy outside. It doesn't feel like 75 degrees. I've even got my, lumberjack thing on here.
Lumberjack.
But I may have to shed it during the course of this. At any rate, we are expecting another rain front. I wouldn't call it a cold front, but it's a rain front coming through this weekend, and then another the week after that. It's all sorely needed. This one may fall on the hill country, which would be great. Fill up the reservoirs, or at least fill them up a little bit. It's all good.
Also joining us from Montreal, Angela Mingarelli.
Hello, everyone. So it is also, cold, quote unquote cold. It's terrifyingly warm for February. It's 41 F and 6 C, I think. The fact that it's above zero in February in Montreal is so troubling. And then everything is just icy and wet because tonight it's going to go below zero again. Everything's going to freeze. And then next week, I just looked at the weather forecast. It's supposed to be minus 17 C in like 5 days. So it's going to dip again.
Ouch.
Yeah, Vincent was right. Winter's coming back.
It will. Yeah. It'll be back. It's February's early.
We'll have snow until April, probably.
Right.
If you enjoy these programs of Microbe TV, TWiV, and all the other science podcasts, please think about supporting us financially. We are a nonprofit, Microbe TV, so you can, in the U.S. anyway, you can deduct the donation from your federal taxes. But also, it's how we function. We have a lot of costs associated with these productions, so we need your support. And it tells us that you want to hear real science, not fake science. And, there's a lot of that out there right now. We're trying to counter it in a very calm way, just by telling you all about science papers and that sort of thing. So microbe.tv/contribute. Don't forget about Viruses of Microbes 2024, which is happening in Cairns, Australia. I was taught how to say that. I wanted to say Cairns like everybody else, but it's Cairns. You drop the I and the R.
No kidding.
Why makes you think, why didn't they just call it Cairns?
So should I have called John Cairns? John Cans? Everybody else called him John
he was
Cairns.
a Brit. I don't know if it's the same thing.
They don't even call it Australia. They call it Strine.
Strine?
Strine.
And what's that from?
That's their accent. It's a bastardized Cockney accent that they have adapted throughout Australia. And
you're going to get
the
mail.
good day and the Aussie, and all these other great expressions. So I'm going to get a lot of mail. It's okay. I spent 6 months down there, and I can tell you that It took me that long to figure out what they were talking about in some cases, but
Jeez, he doesn't stop. The mail keeps piling up.
I
No,
love the
that's okay. I
Australian
don't mind.
accent. I
No,
love
no,
the
no.
Australian accent.
So do I.
have to say.
I do too. I think it's
Yeah.
it's very, very charming.
I
It's,
find the
there-
Scottish more difficult to understand.
Oh,
Like
well,
I
Have you
had
seen
a brother. My brother-in-law was from Edinburgh, and when I met him, he like
right.
said something. I had no idea what he said, and I just smiled and said yes. I was like, yes.
Have you
And
seen the
he's like-
Robin Williams video about going drinking with a Scotsman?
No.
No.
Oh, it precedes his video about golf. I'll send it to you. It's a
I would
riot.
love it. I would love to see
Yes,
that.
please.
I understand
Sorry,
there's good
I
diving
interrupted you.
down there in Australia.
Oh
Angela.
yeah, the Great Barrier Reef.
Yeah, that's where Cairns is. And if you go to this meeting, you can check out the Great Barrier Reef and the rainforests on the other side.
You see, that's where the barrier reef is. It runs the entire length of Australia. It's the
Yeah.
largest living organism on the planet. So you said that's where the proof is. It's also where Sydney is. It's also where. What?
All right.
You don't like that?
It's okay. It's fine. I'm trying to get through this little announcement here. So this is a viruses of microbes meeting. All right, so if you're a microbe, a lot of bacteria, archaea, but also protists. The viruses that infect them are the topic of this meeting. So we're going to go down there and do a TWiV. Jolene Ramsey will be there as well. She's going to the meeting, so she'll be on the TWiV, and we'll have some of the speakers as guests. But if you want to go to the meeting, if you're a scientist, abstract submissions close 5 p.m. Australian Eastern Standard Time, which is UTC plus 10 on Friday, February 16th. Early bird registration deadline is Friday, May 17th, and you can go to vom2024.org to find out more about that meeting. Also want to tell you about a research assistant position in Amy Rosenfeld's lab at the Center for Biologics Evaluation and Review at the FDA. This is to study enteroviruses, immune responses to enteroviruses, animal models, and so forth, all BSL-2 conditions. And you know, the enteroviruses are among the most interesting viruses in the world, so you ought to go work there. If you go to the show notes, microbe.tv/TWiV, you'll find a PDF that has more information and a link to Amy's email. And don't forget to buy Dixon's book, The New City. Go over to despommier.com, and there are all the links to the major booksellers are there. Picture of Dixon chilling, and give him your support. But more importantly, it's a very interesting book. It's getting good reviews, right, Dixon?
Right, Vincent. In fact, I just got a good review yesterday from the American Library Association, so I'm
Cool.
very rewarded for all of the hard work that all of the people associated with that book did to make it look as good as we did. It's amazing that it came out as well as it did, and I'm glad it did. It's one of the
It's
few.
not amazing, Dixon. It's you. Come on.
No, but it's also the book producer. Thank you for that, Richard. But I didn't design the book. I just wrote it. And that's the funny thing about being an author. Not only do you not get to design the book, you don't even get to title the book most of the cases.
I didn't know
The
that.
publisher
Really?
titles the book to optimize sales. And no one had an argument with this one because it was pretty obvious what we should call it. But for other books, they've really bent the nose of the author by changing the title of the book to something that the author did not approve.
What were some of the other candidates that you had as titles? Did you have any?
No, I don't. I wish I had a list, though, because
Oh.
I know I didn't have any. This was my only title. The same was true for The Vertical Farm. Someone said, what are you going to call this book? I said, well, it's all about vertical farming. Why don't we call it The Vertical Farm? Gee, that's inventive. And, it stuck. So,
Well, it's like TWiV. We come up with titles right at the end.
well, we try. Alan does it the best, though.
Actually, the one from last week's just blew my mind. What was it?
No, it was
when
great.
when macrophages go to elementary school? Give me
That's
a
right.
break.
That's right.
Unbelievable.
Good stuff.
Yes.
Good
It
stuff.
takes a certain mind. Yeah.
Yeah.
Yeah. Oh, by the way, folks, do you guys know what kind of bee lives in the United States?
Uh-oh.
This is a joke, by the way.
I saw this coming.
I always know. I
Note,
already know the
Vincent,
answer.
what kind of bee lives in the United States?
That's right.
A U.S. bee.
Oh my gosh.
Is that a dad joke because it's so corny?
That's like a grandfather joke, I'd say.
Oh,
That
I'm
hurt.
joking.
That-
I'm
except,
joking.
that your grandfather didn't know what a USB was.
That's true.
True.
I also saw this Instagram this week. It had a picture of a double-stranded DNA and a single-stranded RNA.
Yeah, that's right. I said it too. That's right.
And the double-stranded DNA says to the single-stranded, "Are you single?" And the RNA says, "Always." So I showed that to my class. I said, "What's wrong with this?" And they said, "Well, there's reoviruses that have double-stranded RNA." Yeah,
That's
so they're
right.
not always
Mhm.
single.
That's right. That's right.
Maybe that
That's right.
particular RNA is always single, but not RNA in general. Okay, let's do a couple of things in the news here. First is a paper in JAMA Network Open. Contribution of infant rhinovirus bronchiolitis to hospital bed and ventilation use. So bronchiolitis is a big cause of infant hospitalization and ventilation. And Paul Offit always talks about how respiratory syncytial virus is the most common one causing bronchiolitis. Rhinoviruses turn out to be the second highest cause of bronchiolitis in infants. So this study looks at a cohort of infants hospitalized in Lyon, France. 86,000 visits per year they get to pediatric emergency units. And these kids have laboratory confirmed RSV or HRV, human rhinovirus. And they basically find that the magnitude of our HRV burden is up to 2/3 of that of RSV-related hospital admissions, half of RSV-related days of hospitalization, and 1/4 of RSV-related days of ventilations. So pretty substantial burden caused by rhinoviruses, which people seem to think are harmless, but not for an infant anyway.
So what's the difference between bronchitis and bronchiolitis?
So the bronchi, and Angela will correct me, the bronchi are the two branches that come off the trachea, right, Angela?
Yes.
And then the bronchioles are the little
Tiny
profusions at the end.
ramifications. Yeah.
Okay,
Yes.
before you get to the alveoli.
It's kind of like a tree branch. You have like the thick branch, and then you have little tiny branches, the bronchioles, and then
Bronchiolitis.
Sorry, exactly.
Bronchitis. Gotcha.
So are the alveoli the leaves?
Yeah, you could say that exactly. There you go.
Okay. This helps me too, because I
cute.
never fully understood all that. And itis is just inflammation of. Right?
Yes.
Exactly.
It's
Yep.
interesting to me that you can have, if you have bronchiolitis, can you have bronchiolitis in the absence of bronchitis?
Yeah, that's what I was asking, actually.
It could. Why not? Right.
I mean, I
depends
think
on the
that it's
tropism.
yeah. You could just always say bronchitis if you're not sure if the bronchioles are also affected. But having it would maybe I don't actually know.
Is bronchiolitis like a more serious condition than bronchitis? Or it depends?
I think it depends. I think it's individual to the case. In humans, I don't know. But
Well, in babies, it's a problem because they're tiny, right? They don't have a lot of airspace to begin
Absolutely.
with. So bronchiolitis can be life-threatening. Yeah.
because if the bronchioles, which are smaller, get inflamed, then it restricts airflow, and then it's not
Yeah.
reaching the air sacs, which is the alveoli, as opposed to the major branches are much thicker, and they're even like there's cartilage in the trachea, and then there's just a thicker connective tissue, so it's not as easily collapsed on itself. So the air exchange isn't as compromised.
So that's
It would be
annoying,
worse.
but not necessarily as life-threatening.
Right.
From what I understand.
So one of the take-home messages from this news article is that the therapies, mostly monoclonal antibody for RSV, are not going to wipe out bronchiolitis in the hospital. There's going to be a lot left over from
Correct.
human rhinovirus.
It's exactly right. Yeah. Okay, another paper in the Lancet, Safety and Efficacy of Onradivir in adults with acute uncomplicated influenza A infection, a multi-center, double-blind, randomized, placebo-controlled phase 2 trial. This is out of China. And this is a new anti-influenza A virus inhibitor. It targets the RNA polymerase. It's been, tested in animals, preclinical studies, and so this is a phase 2, which was done in China, and basically, it's they say no serious adverse events. So serious means, life-threatening, but 42% of the participants had diarrhea. It's not great.
No.
Of course, this is probably a 5-day treatment or something like that. So, okay, you can deal with it. Anyway, they looked median time to alleviate symptoms. They're giving 200 milligrams twice a day, or 400 milligrams twice a day, or 600 milligrams once per day. And I won't tell you the numbers, but they all decreased time to progression of symptoms. So I don't know if this will end up in the U.S. It has to be trialed in the U.S. before the FDA would approve it. But I hadn't heard of Onradivir, so it would be another one to add to the arsenal. Then we have a interesting article, and I don't think we've ever talked about Oropouche. Oropouche.
I had to Google it. I had never heard of this.
It's in my brain somewhere, and that's got to be.
I'll tell you
TWiV.
why. Because Rico in Brazil, our friend, who is also Kathy's friend, works on it.
There you go.
And he's always told us how it's increasing in frequency in Brazil. And so this is an epidemiological alert from the Pan American World Health Organization, PAHO, saying, keep an eye on it. There's been an increase in Oropouche fever, which is caused by Oropouche virus, which is a single-stranded segmented RNA virus. It's Peribunia viridae. It's transmitted by
a vector. Okay.
By the Culicoides paraensis mosquito.
Look at that. By the what, Vincent?
Culicoides.
Yes.
How would you say it?
No. I thought you said something after that. That's all.
Well, paraensis is the second,
gotcha.
the species. Anyway, It's similar to dengue symptoms: incubation 4 to 8 days, fever, headache, arthralgia, myalgia, chills, nausea, vomiting, sometimes meningitis. So this is an emerging infection, and this is actually an alert out of Brazil.
And it's
Colombia.
from sloths, right? Like the natural reservoir is the sloth.
I was going to ask. Yeah, zoonotic.
I Googled it. I didn't actually know.
Sloths. I bet you like sloths, though.
I mean, sloths are cool.
What's that cartoon where someone goes to a motor vehicle bureau and there's a sloth behind the counter and everything takes forever? Do you remember that? I don't know what cartoon that is. It's so funny because the sloth just takes forever to respond
true.
Every question.
The thing that I find most amazing about sloths is that they have algae growing in their hair.
I was about to say that. That was also my
really?
favorite fact. Yes, because they move so slowly, they grow algae.
Is there a function for the algae? Is it a symbiont?
Don't know. That's a good question.
Dixon, did you know there are things growing in your eyelashes?
True. Demodex.
See?
Yeah.
Yeah.
Do you have them, Angela?
Wait,
Everybody
of course
does.
we all do. Did you also know that humans actually gave it to dogs? So mange in dogs is by Demodex follicularum in
Really?
dogs.
I did not know that.
And it actually came from humans, which is why they have this like very, very aggressive immune response to Demodex or Demodex. You can say either way, whatever. And it's actually from humans. So
I'll be darned.
sorry, dogs. So
what is it? A tick or what? What is this thing?
No,
it's an
it's a mite.
arachnid. It's an
mite.
arachnid, yeah. But isn't
A
it
mite.
have 8 legs? I think it's out of the arachnid family, isn't it?
Oh my gosh, spiders
Excuse
in your
me.
eye.
It's a mite.
So, Angela, if you wear fake eyelashes, what's the consequence of that?
Well, I mean, you can clog the little pores in your, what are they called? The
that's right.
meibomium glands where the tick is also, but it's there anyway. It's not only eyelashes, it's also eyebrows.
Okay.
And so this thing that people get, rosacea, a lot of it is actually because of this extended Demodex infection on their face. And it's
look at
treated
you.
in many cases with ivermectin,
Ha.
which is like, people don't know. And normally I don't know if dermatologists tell them, but part of the treatment in like very severe rosacea is ivermectin because of these like parasites on your face.
So does that get rid of all the mites in the pores along your eyelashes also?
I guess, I don't know if they do it in humans. I don't know if it's like topical just to certain regions or not,
I
but
see. Okay.
Cool.
Boy, are we having a good time today. This is great.
You know,
This is TWiV on steroids.
We have a case of measles in Maryland. This is a person an international traveler, or a Maryland resident with international travel, and this is interesting from the Maryland Department of Health. They tell you where the person was, so in case you were there at the same time, you might want to do something, make sure you're immunized. Like Washington Dulles on January 27th from 6 a.m. to 9:30 a.m., and then at a hotel, and then in the emergency department. The patient probably went, so I bet the patient was not vaccinated. There's also a case in Ohio. I see more cases first measles case in Ohio of 2024. This is in a child. Do we know if the child was unvaccinated?
I could not find out, and it would help to know how old the child was. That is, whether they were young enough to have not yet been vaccinated. But I couldn't find
Rich,
I'd like to know what part of
Rich
Ohio also.
probably knows this. What is if there is one specific, more or less, the transmission radius for measles when it's aerosolized? Is it known? I mean, I know it can go in the wind and it can be up to like X amount of, but is it like, is there a generalized
I don't
distance?
no, I don't know. My only reference for this is one of my ex-students used to work at the CDC on both measles and mumps. And what he was fond of saying is that if you're sitting in a room unvaccinated and somebody with measles just walks through the room, you're going to get measles.
Wow.
Someone told me that if you're in a lecture hall and you're lecturing and you have measles, the people in the back row will get it. It's like 100 feet or something. It's a long ways.
Yeah.
I'll say.
All right. This is a very interesting article in JAMA Network Open. School vaccine coverage and medical exemption uptake after the New York State repeal of non-medical vaccination exemptions. So did you know that in 2019, the New York State legislature eliminated non-medical vaccine exemptions in the state?
I mean, like religious exemptions.
Yeah.
Whatever you want. I don't believe in it. I'm not putting anything foreign in my body belief stuff, right?
Oh, that's
only a medical, which is totally acceptable. A medical, of course, there are many people who cannot be vaccinated.
Mm-hmm.
And so they did a survey to see the effect of that on vaccination coverage, right, overall. And so the bill, and this is, of course, an association, you don't really know, but the implementation of the bill was associated with an increase in mean vaccine coverage of 5.5% among non-public schools and 0.9% among public schools.
That means to me probably that the non-public schools had a higher vaccine exemption rate to start with relative to the public schools. So you saw, they had a different background. So
Also a slight decrease in medical vaccination exemptions because a lot of those are bogus, right? They're just not true. They're way higher than they should be. And if you're wondering why New York State passed this law, there are a couple of big measles outbreaks in the state, and it was in communities that do not vaccinate. And so they said, no, this is unacceptable. And so there you go. And this is a measure of the efficacy. It's not a lot, right? But better than zero. Then there's an article here that doesn't work. The link doesn't work. It's at a website called pig333.com, which I think
mine
is
opened. Mine
Yeah,
opened.
I had this working.
Yeah.
It was working for me.
It didn't work for me.
You
I
must
get
have a
forbidden.
firewall.
I don't have permission to access.
Oh, my goodness.
So what tell us is about foot and mouth in Europe?
Yeah.
No, mine opened. Mine opened.
Increased risk of foot and mouth disease entry into Europe. The increase in number of outbreaks in several North African countries and recent detection of the SAT2 serotype in Algeria increase the risk of disease entry.
So
Which could be
the,
disastrous. Foot and mouth
yeah.
disease getting into
Right.
Europe. Yeah.
So it's a big problem in cattle, right? Does it affect any other agriculture? Does it affect pigs, Angela?
I believe so. I know it affects other ruminants like sheep and goat. I don't remember if it affects pigs. I think so, actually. Yes, I think so. I'm remembering images of pigs in vet school that with the vesicles in their mouths. Yes, it does. Yeah.
Okay.
All farm animals, as far as I'm aware, pigs, sheep, goat, cows, and it's extremely contagious. And as of like the OIE guidelines, if there's any sort of foot and mouth disease in any farm or even anywhere near another farm that has had it, they have to call all of the animals. It's complete,
right.
there's no trying to treat them or anything. It's very,
So in the U.S., we don't allow research on we don't have the virus, and we don't allow research on it except at Plum Island at the end of Long Island. And that facility is being transferred to Manhattan, Kansas.
It is?
Yes.
Oh, I didn't know that.
Oh, my God.
Yeah, there's a new facility in Manhattan, Kansas, and the Plum Island works are going to go out there. And
Wow.
they work on like foot and mouth and other
Rhinovirus.
high-risk pathogens for animals, not for people, but for
That's
animals.
right. That's
yeah.
right. That's
Yeah.
right.
But
According
imagine
to
if it
Wiki-
got into the cattle in the United States, the amount of cattle and just the economic
man.
implications
No,
of
it'd
that
be a
would be
disaster.
Horrible.
devastating. Yeah.
They also worked in Hamilton, Montana on that too, don't they?
I don't think there's an agricultural virus unit there.
Really?
I don't believe so. I think it's just Plum Island, nowhere else.
I think that's right.
I see.
So according to Wikipedia, the susceptible animals include cattle, water buffalo, sheep, goats, pigs, antelope, deer, and bison.
Wow.
So it's basically all ruminants and pig.
Right.
Did you say horse? No, not horses, I believe.
I didn't say
It's
horse.
mostly
It's also
it's all
been known to
ruminants.
infect hedgehogs and elephants. Llamas and alpacas may develop mild symptoms, but the major ones are those I mentioned.
Okay.
And people know.
No.
But
No,
we are
people
very
don't.
close to pigs in lots of ways. It's I don't mean our appetites, but I just mean our physiology. It's interesting that we avoided that one. That's good, I guess.
we take small win for us, but
Right.
take what we can get.
That's right. That's right.
For our snippet today, a Nature Medicine brief communication, severe disease during both primary and secondary dengue virus infections in pediatric populations. Now, we've talked about dengue a lot, but remember, this is a mosquito-borne illness. It's borne by the Aedes aegypti, so it's only in places where you have that mosquito. And it is there are 4 serotypes that, when you are infected with one of them, cause similar to Oropouche fever, high fever, headache, muscle joint pains, nausea, vomiting, and so forth. And the idea has always been that the first time you get infected with dengue virus, it's kind of a mild illness. But then if you get infected a second time, you can get severe dengue, hemorrhagic fever, and die possibly. And it's called, antibody enhancement, because we think the antibodies to the first serotype do not neutralize whatever serotype you get Next, they rather facilitate uptake of the virus into, say, macrophages that are not normally infected. By the way, we had a guest on TWiV many years ago, and she's at Hopkins, Nina Martin, and she had been working somewhere as a volunteer and got dengue, and she talked about it. She said it wasn't so mild; it really hurt. So anyway, this paper looks at this question: is it really? So we have primary dengue the first time you get it, and then secondary after that. Are they really different or are they similar? So this is a study out of India where they have more dengue than anybody else now. And they were able to go in hospitals and look at medical records. And in children, 2 months to 16 years of age, ask with confirmed dengue in different hospitals in India, what is the severity of the disease? So they have 3 different hospitals in different regions, ages of the children 2 months to 16 years. They have, in 65% of the patients, they identified the serotypes. So they had both 1, 2, 3, and 4. And then they classified them as either primary or secondary based on the ratio of IgM to IgG, right? Because if it's a secondary infection, you're going to have IgG. You're not going to have IgM. So that's pretty cool. So they have a mix of primary and secondary infections. They have 344 primary and 275 secondary out of 619 infections that they looked at. And they determined severity using WHO guidelines. And they find that the frequency of severe disease is the same in patients with primary, 32%, or secondary infections, 32% in both cases. So that's, they said this surprised to them. And they saw it at all 3 clinical sites. It's not associated with particular dengue serotype. And so they tried to apply more stringent classification of primary and secondary. Still came out the same. A lot of severe primary cases. Didn't vary according to age. They addressed this hypothesis that this is one idea that, the mother gives antibodies to the child that last about 6 months, right? Mm-hmm. So they're saying, well, maybe kids get up to 6 months, maybe they get severe dengue because of those. But all the kids that had severe dengue in their study, none of them had detectable IgG antibody. But they did have dengue-specific IgM. So it's not because of the mother's IgG. These infants also had very low neutralizing antibody responses as well. And so they did look at the neutralizing antibody responses in these kids. And the ones who had primary infections had lower neutralizing antibody.
There's barely any compared to the secondary, right?
Mm-hmm. In the secondary, you would expect to have more, right?
Yeah, but the disease outcome was similar, which was interesting.
Yes, very interesting. Also, kids with primary had a narrower breadth of the neutralizing antibody response, which I guess you might expect as well. And they say, okay, so that's the finding, but they say other places have found something different. Other historical studies say that most of the severe dengue are secondary cases, but we are not. We are not seeing this. You see it in, and they say, we don't question that ADE, antibody-dependent enhancement, can occur, but it's possible that in a primary dengue infection, you also get severe disease as well. So I think this is a very important finding because that I teach it. It's saying, all the severe dengue is ADE, but it's not.
Yeah, this is a paradigm shift.
I think so.
Okay. It's like, you got to, this is one where you got to go change all the textbooks and change your lectures. Okay.
Yeah, but then they don't really discuss how they think this is happening. I guess it's still an interesting finding, but where does ADE stand then?
Exactly.
secondary infections in being more or less lethal, not lethal, because I guess lethality is very low, but like disease, like severity.
What? So, I mean, I guess the secondary infections here that are severe, though, some of those could be ADE, or maybe all of them could be ADE, maybe, and then the primary are not, right? By definition, they can't be because there's no memory response being made.
Mm-hmm.
So, or here's the thought, folks. Now they're kind of young, these kids, right? There's no stratification by age, but I was thinking maybe they had another infection and their antibodies are cross-reacting. And we don't know about that. But, I mean, 2 months and up, what was the lowest age here?
It was 2 months, I think. 2 months to 16 years.
I guess you can have an infection, but you're not going to make your own antibody response, right?
Probably.
Actually,
maybe you
yeah,
had-
it looks like the oldest was 16 years. I wonder if you move out into an older population, if
They said that
things
didn't
look
make any
different.
difference.
Didn't make any difference.
No,
Okay.
no difference in severity. So I think I don't know. I think it's still possible there could be cross-reactive antibodies that are causing ADE in the primary infections, and they're not dengue antibodies. They're something else.
Well, this ought to spin off a lot of other studies that try and figure out, okay, what is ADE? How prevalent is it really? You know, to sort of
Yeah.
reevaluate
Yeah.
all this stuff.
Also, we need a good dengue vaccine, and there are other. Dengvaxia is not good enough because it causes ADE in some kids. So we do need a new one, and at least one is under development, and others as well. So that will happen. Aedes aegypti. Dixon, is that a peridomestic mosquito?
You bet.
Okay.
Yeah,
I know
it's
the words.
extremely well adapted.
Yeah. And it is is it a night biter, or
No, late afternoon.
what is that called? Crepuscular?
Yes, something like that. That's
Look
it.
at you.
I've been listening to Dixon for
Oh, well, I don't
how
think
many years?
I
How many years have I been listening to you?
I don't think I've ever used that word in public.
Crepuscular?
Crepuscular. The mosquito, though, is a tree hole breeder, and it breeds in temporary bodies of water that makes it very effective, not that time, but the next time it rains. And that's the tricky part of this mosquito. You can actually collect the eggs in laboratory-reared mosquitoes and save those for years in a refrigerator and just take them out, put them into water, and they hatch. It's like instant mosquito. And, you can imagine some interesting plots that you could write about in a science fiction novel about the genetic engineering of Aedes aegypti to transmit all kinds of lethal diseases. And then you distribute them with your secret agents, and they all drop it into a little puddle of water, or they go into a church and throw it into the holy water. I've had these horrible nightmares about the ease in which it is to start a colony of Aedes aegypti anywhere, basically. So you really have to survey the area for tin cans and containers that are left unattended, and boats, tires.
Is the
were
vector
used.
for Zika also Aedes aegypti?
Yeah.
It is not, and it's also a flavivirus as well. Is it known if there's cross reactivity between Zika specific antibodies and dengue?
Yes.
Do we know that?
Yes, there is.
Oh, there are.
Eva Harris told me, I saw her at some meeting recent, ASTMH, I think, and she said they had a study in Nicaragua which showed that, yeah, they have cross-react. I was just looking at the U.S. data.
Mm.
There were 54 travel. How many? There were a bunch of travel-associated cases of dengue in the U.S. in 2023. 27. So people get infected at somewhere else, and they come to the U.S. and then they get diagnosed. But there are also some locally acquired ones in Puerto Rico because they have Aedes aegypti.
We got local dengue infections in Texas.
Texas, I
That's
think,
right.
yeah,
That's
probably
right.
not
Wow.
You
2023, yeah, because I
do.
remember that. Texas
do.
and Florida also, right?
My
Yeah.
one of my yoga teachers is an epidemiologist in Texas, and so she's my resource for what's going on.
You take yoga?
I do, of course.
You take
yeah.
yoga. Do
You
you?
go
Yeah.
somewhere and take yoga?
Yeah, I mean,
I can see Condit with his little mat rolled up, and you have
amazing,
tights on.
Rich.
Hey,
Do you
my
also
wife-
have tights?
My wife and I are in the our local YMCA is awesome, and they have all sorts of
great.
different classes. We're there every day, and
Right.
some of the classes we take are yoga classes.
What does it do? Does it make you more flexible?
Yeah. Oh, it's great. Stronger and more flexible. Okay.
Do you do that, Angela?
I've done yoga as well. It's not like my main mode of exercise. I do weightlifting more than yoga and like swimming and things. But yes, exactly. Olympic weightlifting. Yes. I like to lift weights. But hot yoga is actually really fun too. I
I'm
don't know
not
if
doing
you've
that.
done it. You
I'm not
haven't
doing
done
that.
it.
Bikram, that's Bikram.
I'm not going
The
there.
Bikram, yeah. It's an
I'm
hour
not
and a
going
half.
there.
I think it's like 45 degrees Celsius.
It's
Something
very
like that.
warm in there. Yeah.
It's
And
hot.
the room stinks probably too, right?
Well,
Well-
if you go to a good ventilated yoga studio. Well ventilated, I should say.
Okay.
Good for balance too. And
Yeah.
That's true.
as the mature individuals on this show will note, one of the first things to go is balance.
Tell me about it. Tell me about
Yeah,
it.
I know. I know.
That's right.
I people who have done yoga for many years, they have a way of walking which is distinct. It's very balanced and beautiful. Their posture
This is
is
true.
really good.
It's very true.
It's very.
Nice
I
and
wish I could
toned.
do it.
You could.
Wish
Why
I could
can't
do
you?
Yeah, you
Go.
can do it. You
Do
wouldn't-
it.
Well, what am I going to do? Take an hour at noon to go do a yoga
Why
class?
Yeah.
not?
I'm sure there's
Exactly.
many around you. They're everywhere
There
in New
is.
York.
There is.
That's the deal. Okay.
Ah,
You actually make the time to do this stuff.
Vincent's thinking
you'll
about
come
it.
back a new man.
I would really like to because I'm getting less flexible and I
There
don't
you
like
go.
it.
You should do
right?
it. No, you should do it. You
And
should
it
do
doesn't have to all be yoga. Find yourself a gym that has all sorts of different classes and go. You know,
No, that's-
I
It doesn't
do.
have to be yoga.
At
So
least-
what else is there?
come on.
There's a thing that we
Pilates?
do. There's Pilates. There's a thing we do that I do twice a week. It's a internationally distributed thing called body pump.
Oh,
That's
yeah.
an
Body
that's
pump.
an
Yeah.
hour of low weight, high rep free weights mostly. Okay, that goes through and works every major muscle group, and my whole deal, our whole deal is you just got to keep moving, man, or you're
That's
just
true.
going to stiffen up and it's going to
Yeah, of course.
you're going to lose it. I
Keep
swim.
your blood flowing.
I
keep your
swim
Well, I
at
do
the
other-
gym.
I do exercise, but I would like to work on flexibility.
We got a thing called Senior Fitness. That's the brainchild of, I think, the instructor in this, who used to be a dancer. Okay, and it's basically movement and stretching and weights for old codgers like me, and it's really really good.
All right.
Really
So
good.
can I get into a beginner class where nobody's going to laugh at me? Because
Nobody's
Don't worry
gonna
about it.
laugh
Nobody's
at you.
gonna laugh at you,
Nobody's gonna laugh.
Vincent.
You wouldn't believe the population of people you'll find in these classes. Every age, every body type is there.
hmm
Go for it.
La-
Forget about
laughter
the
is-
other people.
Laughter is very healthy for you, though.
I just want to find someone some place I could
Yeah.
walk to from the incubator. you know?
Exactly.
tons
That's
of them.
exactly
No,
what you need. There
come
got to
on.
be.
Maybe
a
there's
place.
one in this building. Who knows?
Could be.
Vincent, I'm convinced that you live near one.
Oh, I'm sure.
That you,
No, I
that you
don't
work near one. Work near one.
work. I want to find one here. I will do it.
And my insurance, my health insurance, pays for my YMCA membership.
Wow. that's
Everybody wins. The health insurance company wins because I'm more fit. I win because I'm more fit. And the gym
YMCA.
wins because I'm going to the gym.
cool. Very cool. All right. Now we have our paper, which is fascinating paper. Well, we could stop now because I'm.
No.
No, we don't want to
Go
stop.
find a
Another
yoga
TWiV
place.
is viral, right? Yeah,
I
we
mean,
can stop.
you
You
could
got to go to the
also-
gym.
You can also just go to the gym if you don't want the community based in a class. You can also just go to the actual gym. There's lots of like just to do weightlifting or to do machines, or if you want to be independent and put on your AirPods and not have to like interact with others. It's up to you. There's different
get
styles.
yourself a
Interact with people in yoga.
Well, I mean, if you go to a
No.
class, you're normally like there's other people around. You can choose to, you know, talk to them, get to know
That's
them.
fine. I don't mind. I
whatever.
like talking. Did you know, Angela? I like talking.
I've noticed. Yeah, I've noticed.
No problem with that. I could probably get some more TWiV listeners in the yoga class.
That's right.
True.
You can get yourself a personal trainer too if you want. That's another way to approach it.
They could come here.
No, but
Nah,
you
you
should
don't wanna
leave
do
the
that.
incubator. Yeah,
you got to
no,
leave the incubator. You got to go somewhere, Vincent.
Okay. I'll leave the incubator. Well, actually, one of my sons is a personal trainer.
There you go.
He has been doing weight training with me once a week.
Okay, good.
Okay, good.
Nice.
And it's really good. We do, deadlifts, squats, and bench press. That's all he said. This is all you need. And he just does an interesting program where he mixes it up, and I like it. very much.
Powerlifting. Yeah, the main compound movements: bench, squat, dead. That's what people say.
Bench, squat, deads.
I'm going to the gym after this TWiV actually to squat. I'm gonna go.
just to squat, nothing else.
Well, that's like my main, because you always have like one main lift and it's gonna be really heavy. And then after that, I'll do accessory work. Like I'll do other things, but I start with squatting because it's the hardest on your central nervous system. Literally though, like it takes a lot of strength because
Really?
I like lifting heavy.
So he has a belt, because he does serious. So he had me wear it the other day just to feel the difference. It really makes a difference when you don't feel that your intestines are gonna.
Did you learn how to brace? How to brace
Yeah.
properly in your belt?
Yeah. Brace he's teaching me. He's very good with it. Yeah.
Good.
Anyway, the paper is Nature Communications. Therapeutic mitigation of measles-like immune amnesia. And you guys just want to talk about exercising?
I could talk about the gym for like 5 TWiVs. I could talk about the gym all day. I could get you on creatine in 5 minutes. Like,
what do I need creatine for?
To maintain muscle mass. 3 essential amino acids, 5 grams a day. You're good. I take creatine.
Wait,
Everyone I know
creatine
does.
is not an amino acid. It's a
No, it's 3. It's 3 essential amino acids. I think it's like proline, arginine. I forget which 3. I have to Google it now.
Yeah, my son
fact-checking
takes
me.
creatine. I know because an order always comes every month from Amazon with a big bottle of creatine.
Exactly. Yeah, glycine, arginine, and I don't know which other
So
one.
you take creatine,
Yes, I do. Yes.
and do you notice it has an effect?
I noticed that overall strength, I have like more energy at the gym in my lifts, in my heavy lifts. I do notice it.
You really could talk a lot about exercise.
Oh, I could talk about exercise. It's like my other like freaky thing that I love to learn everything about. I consume so much YouTube content on like bro
Exercise?
science, but like actual science. Like, yeah.
Oh, cool. Well, listen, come back on office hours. We'll talk about exercise.
Oh, we should definitely. Yes, we should definitely. You should have your son on as well. The 3 of us.
Well, I don't think he'd want to come on, but
You can ask him. You can
could
say
ask
that
him.
I exercise and we can all talk about, all of this.
yeah, we can have a special
Weightlifting?
episode on exercise. Yeah, nobody will come because they don't want to hear about that. They want to hear about viruses, but
I think you'd
It's
be
okay.
surprised. People like
Yeah.
talking about exercise, especially if we
didn't
get
finish
into like the nitty gritty. Sorry, the title. Back to the paper.
the title, the therapeutic mitigation of measles-like immune amnesia and exacerbated disease after prior respiratory virus infections in ferrets. So really interesting. So the 2 things here that are important. So measles virus, respiratory, we just talked about outbreaks of measles, right? It's vaccine preventable, but if you're not vaccinated, the normal pathology is that you inhale the droplets from someone else. It goes into your lung, reproduces in immune cells. Those cells get into your circulation. They produce virus, makes a viremia. It goes to the skin, you make a rash, but it also reproduces in memory T and B cells and messes them up. It erases your immune memory. And that's work that Michael Minna published a couple of years ago. We had him on immune to actually talk about it before pandemic. And that's really bad, right? Because if you lose your immune memory to all your vaccines and any prior infections, you need to repeat them all, right? And you're going to get opportunistic infections. And this is a real problem. So,
So
this is one of the-
even if you don't get, if you get a measles infection, even if you don't get like severe disease or anything like that, it's going to erase your immune memory, right?
That's right.
Man,
That's right.
That's serious business.
Probably, I mean, measles can do other bad things. It causes encephalitis in 1 in 1,000 cases, right? It can kill you if you're malnourished. But erasing your immune memory is really bad. And I don't see why anyone would say, I'm not vaccinating my kid, because that is a real serious thing, right?
Yeah.
So this is one thing. So they ask in this paper, could we use an antiviral to somehow, if you do have someone who is not vaccinated and they get infected, could we treat them and prevent immune amnesia? So they use an animal model to do that, by the way.
But we haven't talked about the authors and affiliations.
Hold on. I want to show you the animal model that's used.
Yes.
It's
Well,
the
fair.
ferret. Okay. Affiliations, that's right. Well, are there co-first authors? Because you have to go to the end to find out. Does anyone know?
Isn't it Robert M. Cox and Joseph D. Wolfe?
Could
They're
be.
the two co-first.
Maybe. I don't know, because
And the last author is
Richard Plemper.
Plemper and also Greniger, Alexander Greniger. I think
So
they're...
Plemper's been on TWiV a couple of times. So
And we
talking
did a
about
paper
measles.
where he was an author just last week, right?
Oh, that's right. Yeah.
I mean, this
Yeah, which
this
was-
basically is continuing some of the discussion we had last week.
Mhm.
Yeah, that was about the macrophages that go from the from the intestine, elementary school, from the intestine to the lung, and they'll have a role here actually in this paper.
Right.
And the second is, what about if you have multiple respiratory infections? What happens, right?
I don't know
another
if we said this
respiratory-
is from Georgia State and University of Georgia and other
Right.
institutions in Georgia. Okay.
I thought that this paper was so cool when I was reading it this morning. I got like extremely excited about it, and I don't know why. Just everything. I thought it was very well explained.
That
And
was
it was
beautiful.
like the experiments were like easy to follow. The way it was written, everything about it, I thought was really, really good.
It's a
I
good,
totally
it's a
agree.
good paper. Yeah.
Mhm.
So the one of the things that Plemper talked about when he was on TWiV, what's the episode number? 636. I visited Georgia State, and he said, we have an antiviral against measles, but nobody wants to use it because the vaccine is so good. Well, now this paper shows why it would be useful. So they originally had this polymerase inhibitor called ERDRP0519, which in ferrets, it protects them against a lethal infection with canine distemper virus, which is a related virus. So, Angela, this infects dogs, canine distemper? Does it cause canine distemper?
Yes,
Yes.
exactly. It's part of the regular vaccination that your dogs get when they're very young. I think it's at 6
Right.
weeks. And then there's multiple vaccinations, actually. I think it's 3 separate vaccines with a couple weeks apart until 12 weeks. 6, 8, 12, I believe.
Okay. So ferrets can be infected with canine distemper, CDP, and they get disease, but you can't infect them with measles. So CDP serves as a model, and that's what they use in this paper.
By the way, Vincent, just as a side note, the distemper virus vaccine was developed at Cornell University on the campus that you went to. Yeah, that's right, in the vet
New
school.
York. Cool.
I want to take a moment to very briefly do the pathology because it's relevant to this whole thing. So, first of all, we're talking about a negative single-stranded RNA virus enveloped. Okay.
Right.
Correct me if I get any of this wrong, but it's acquired as a respiratory infection, and the first target is macrophages and dendritic cells in the lung.
Yeah, alveolar macrophages.
Alveolar macrophages, our old buddies from last week. Okay.
Cyclic F positive macrophages. Yes.
And then it winds up establishing a viremia. That is, it spreads through the body in the blood system because it's infecting these white cells. Okay. And ultimately that circulation returns it to the lung. And now you're on the other side of the alveoli. What would that be? The basolateral surface. Okay.
Ooh,
And
that's impressive.
now the virus released from the macrophages infects the epithelial cells. Completely different receptor, completely different cell type, and trashes the epithelial cells. And that, I think, is responsible for a lot of the disease pathology, plus the immune response to that that you see. And am I correct, Vincent? That was Roberto Cattanio's work, figuring all that out.
The receptor on the basolateral domain. Yeah, it's a different receptor from the one that the viruses get into the immune cells at the top. Yeah.
So to me, that's a really, I don't know if it's unique, but it's a very interesting pathology where there's two very distinct classes even of cells involved, starting with white cells in the initial infection.
Right.
That helps circulate and you wind up in the epithelial cells. And that becomes important for this paper.
It's interesting that on the way in, the virus does not infect epithelial cells, right?
Right.
It infects immune cells, but on the way out, it goes in.
How does it get into the macrophage then?
There's a different receptor.
No, how?
What do you mean how?
Doesn't it have to
phagocytosis. Yeah.
Doesn't it have to go through the epithelium to get to the macrophage?
No, the macrophages from last week. I know this. The macrophages line the alveoli.
Well, they're on the air side of the alveolus.
Yeah, they are.
And then okay.
But they could go back and forth. They can
diapedese.
Diapedese. Is that right? Okay. So the first thing they do is say this. They have a new drug. Actually, they made a different one since that first one. So the first one is ERD, let's say, and the second one is GHP. And they don't have cool names yet because they're not licensed or anything. And so they give this GHP to ferrets, and they just want to figure out the dose. Okay, so they dose them at different levels, and they figure out what dose is results in the drug being present where it needs to be, and also which that concentration will inhibit reproduction in cell culture. This is again canine distemper virus, which could be also inhibited by the same drug. Okay, so then let's do some protection experiments in ferrets. They have these two drugs, GHP and ERD. They compare those to vaccination and to nothing control, and they use a recombinant canine distemper virus that's highly pathogenic. So they infect these ferrets intranasally, and then they give them oral treatment with the drug 3, 5, or 7 days after infection. And then there's a vaccinated group where they have, as Angela said, they have a canine distemper vaccine called PureVax, or Distamink. Do minks get it too?
Yes, I believe so. The mustelids as well. They'd be susceptible, I think. I don't know if it's a different serotype, but...
Is this a mustelid, the ferret?
That is a mustelid, yes.
Yeah, minks are just fancy ferrets, right?
Fancy ferrets, exactly.
This guy wants to be on my microphone. Look. So then they... let's see. So they compare what happens here. Oh, also they give them rabies virus, okay? Because rabies vaccine.
Yeah, they come vaccinated. They got vaccinated before they ever got them, I think.
Right. Okay. So they want to know if this virus will erase their immune memory to rabies virus, basically.
So they tested the antibodies before against rabies virus and then after. Yeah.
That's right. And they look for signs of infection. There was fever, rash, loss of body weight. They look at viremia. They look at complete blood counts because the infection is going to reduce the white blood counts. And as Angela said, the titer of anti-rabies neutralizing antibodies. So the animals that didn't get anything, they got vehicle, they died within 12 days. The GHP antiviral worked really well. It's complete survival, reduced viremia when they gave it at 3 or 5 days post-infection. But 7 days, not so great. And the other one, ERD, the first one that they had developed, even at 3 days, only partially protected. So it's not as good. So they stick with the GHP for a lot of the studies as well. So the vaccinated animals, these are prime boosted animals, they are protected. But if they got vaccinated a day after exposure or a day before exposure, didn't protect them from clinical signs, which you would expect, right?
I think there's this idea that has been going around that vaccination can be used therapeutically somehow.
Yeah, right.
And
right.
so this is an effort to test that, and it didn't work.
Yeah. Right. And also, if you vaccinate them 2 to 3 days prior, that doesn't work either. So you need more time.
Right.
Right. Let's see. So lymphocyte counts drop. That's why they're measuring because the virus is reproducing in them. That drop was prevented by antiviral treatment. Both drugs at 3 days prevented drop in lymphocyte counts, which is called lymphocytopenia. The GHP at 5 days didn't prevent this collapse, but the white cells recovered within 4 weeks. And remember, these are going to be important for memory, so that's why we're looking at that. Fully vaccinated animals do not develop lymphocytopenia, interestingly, right?
So this may be obvious, but this is one of the reasons the pathology is important, is that the first target of this virus is lymphocytes.
Yeah.
Okay. And so in the process of this whole pathology that I talked about, a lot of those lymphocytes die, and that's why the memory disappears. Right? If I got that right?
That's right.
Yeah.
That's absolutely right. Now, what about
maybe as of day 7, because then we see if it's a post day 7, then there's no recovery, but maybe too many lymphocytes have died by that point. It's just, or as but previous to day 5, which is kind of crazy how it's a 2-day window or 2 or 3 days, let's say, but it's quite like a big difference, those 2 days within the lymphocytes.
In these small animals, things tend to be compressed, right, compared to humans, especially even in mice, it's even more compressed, right?
How long does a ferret live?
I have no idea.
Like 10 years, maybe?
Really?
Around there? Let me fact check myself.
This one is just 2 years old.
Well, 7 years. Okay.
I must say that I was put off by Figure 3's title. I don't know if anybody else caught that or not, but if you try to read this title to somebody, it's after the therapeutic efficacy of, it's a series of abbreviations and numbers that doesn't make any sense at all. I would skip right past that, except the data below that are so clear that I would like.
Yeah,
they should work a better way of.
Well, they chose not to give
other
easy names to their various
reagents. Understood. It could be vaccine 1 and vaccine 2.
So how about the rabies immunity?
Oh.
Okay, so the antibodies to rabies were preserved in animals treated at 5 days and earlier, but completely gone in one surviving animal who was vaccinated 3 days before, and that animal died. That didn't work. So this drug, GHP, prevents lethal infection. It mitigates lymphopenia when given up to 5 days post-infection, and then allows the lymphocytes to recover or not be destroyed to begin with. Mitigates lymphopenia. Okay, so they use GHP for all the subsequent experiments. Now, it turns out that this canine distemper virus they're using is 100% lethal, which they don't like. So they actually engineer some viruses of different lethality, which is quite interesting. They can actually delete parts of the nucleocapsid protein gene, and they say this is cool. They can make genetically stable, tunable, attenuated virus. So depending on the length of the deletion, between 39 and 55 amino acids, you can have more or less lethality. It's tunable. You can turn a knob and get your lethality. And
they called this portion of the protein an unstructured region. I see it, and I think it's even a tail on the nucleocapsid. So the nucleocapsid is this protein that coats the RNA, and actually stays in association with the RNA while the replication is going on, which is still a mystery to me, is what the interaction is between the polymerase and the encapsulated RNA. But it kind of makes sense that this unstructured region, well, sort of kind of makes sense. Unstructured region on the nucleocapsid, if you tinker with that, might modulate the replication in some fashion that could attenuate the virus. Really interesting stuff.
So they compare these viruses that they make, and they decide on one, which has and that's one of the names that Dixon was objecting to: recombinant CDV N Delta 425479.
Yeah.
So this virus makes a lot of the signs and symptoms. Well, they make a disease in ferrets that they say recapitulates hallmarks of human disease. It's accelerated, right? As I said before, everything is accelerated in a small animal. And infection with this virus, which they made, causes the rabies antibody to collapse. And it takes 3 months for it to recover. So it gives them a window where they can work with their antiviral drugs, right? Because if you remember, the other virus just destroys the white blood cells and they never recover. So that's not so good. Okay, so then they take this virus of intermediate pathogenesis, and they want to know how can we use these drugs to mitigate immune amnesia, right? And so they're going to give the drugs at 4 different times. They're going to give the drug. They're going to infect the animals with the virus, and then give the drug at the first onset of fever, which is about 5 days, at the onset of rash, which is 7 days. And if you wonder how you see a rash on a ferret, you look underneath here, its snout, where there's not much hair, right, Angela? That's the idea?
Mm-hmm. Yeah.
You can see a rash. They have a picture of a ferret with a rash. So this way you don't have to shave them all, right?
Exactly. And you can also see it sometimes on their stomach, like on their axilla, where they have like a little bit less hair. It's
Less
thinner.
hair. So
yeah.
Paul Dupré has made a GFP-containing canine distemper virus that he uses in ferrets, and you can see the lips glow.
Oh my! Really?
Wow.
Yeah, the ferret
That's
lips
crazy.
glow. Yeah.
Wow.
Yeah. They glow green, of course. All right, so onset of fever, 5 days, onset of rash, 7 days, severe disease with diarrhea and conjunctivitis, poor ferrets, 10 days, and then fatality. The first fatality in the vehicle group, 24 days post-infection. And they continue the treatment with the drug until they see antibodies to the canine distemper virus. And so they're going to use rabies immunity because these animals all have rabies vaccine, but they're also going to vaccinate them with a quadrivalent influenza virus vaccine 28 days before infection with canine distemper virus.
So the rationale there, right, is they're going to set up a different immune response and then watch after the infection. They want that as another tracer to see whether you get the immune suppression.
Exactly, because by 28 days, they should have memory T cells, lots of like immune memory.
So, yeah, it's a sort of another data point in addition to the rabies markers.
Yeah. So these animals are monitored for 5 months after being infected with canine distemper virus, okay? And then they're going to be injected with a variety of things, including influenza virus of different kinds. They're going to be injected again with canine distemper virus and also flagellin to look at induction of innate immune responses, the protein from bacterial flagella, right? Okay, so what do they find? So all the drug-treated animals and all of the vaccinated ferrets, the ferrets that were vaccinated against canine distemper virus, they survived the whole study. Half of the animals in the vehicle group died within 26 days, which is longer than with the other virus because this is attenuated, right? Now, you give the drug at 5 days, you reduce viremia. But if you give it later, it doesn't affect viremia. So 5 days is the key. And if you give the drug up to 10 days after infection, you reduce virus shedding into the nasal secretions. So that may be important for transmission. If you treat at the end of acute disease, 24 days, you don't have any effect on viremia or virus shedding. Now, what about lymphocytopenia? That was suppressed in ferrets treated at 5 days, which is, they say, it's kind of like vaccine-mediated protection. And it's alleviated in the 7-day treated group. If you treat later than 7 days, which would be 26 days, the lymphocytopenia is severe and resembles that in the untreated animals. And the lymphocyte counts don't come back up until 10 weeks after CDV infection. This is interesting. Now, they found canine distemper RNA for 64 days in white blood cells extracted from ferrets of all groups except the vaccinated animals and the animals treated at 5 or 7 days. So early treatment or vaccination accelerates viral clearance and prevents persistence of viral RNA, which they say correlates with an earlier appearance of antibodies to the virus. So now they stimulate these animals after they've gotten the drug, after they've been infected with CDV. This is 5 months later. They stimulate them with flagellin, and they monitor a panel of pro-inflammatory markers. And after the flagellin injection, the body temperature goes up, which is what you would expect. And there's no difference in these inflammatory markers between infected and non-infected, which they show the innate response to an immunogen is not compromised after recovery from acute CDV disease. All right, the innate response. Now, all these animals had antibodies to rabies and influenza virus, which they had given them a vaccine against. If they treat the infected CDV-infected animals at 5 days after infection with the drug, you preserve neutralizing antibody titers against these two viruses. Okay. If you treat later, you have a rapid decline in antibodies to rabies virus and antibodies to influenza virus, which is similar to the vehicle. So again, as Angela said, you have a 2-day window to prevent the decline in memory. It's really crazy. Interesting. Let's see what else is. All right, so that's a cool result there. That the drug at 5 days will prevent antibody decline. Now, what it would do in people, if it works at all, we don't know. You have to do a clinical trial, but maybe you have more of a window in people than that, but we don't know. They wanted to look at the effect on the cell-mediated immune response. So far we're looking at antibodies, right? So they infect the animals with a different influenza virus subtype, H3N2. They were vaccinated against a different virus.
H1N1, wasn't it?
Yeah, they were H1N1, the pandemic, 2009 pandemic, right? Mm-hmm. So these what they find is that humoral immunity to this new to H3N2 collapsed in all the other groups except the groups treated with the drug at 5 days after infection. None of the groups had clinical signs. Antibodies came up within a couple of weeks. Low levels of virus shedding. So they're saying there must be some cell-mediated immunity, cross-reactive immunity from the vaccine, the 2009 vaccine, or challenge actually, which is protecting against H3N2 because there's no antibodies there. They've been destroyed by the canine distemper virus infection. So that shows you that antibody, cell-mediated immunity is very important for recovery, at least in ferrets, right?
Of course. I mean, we always forget about T cells. We're always talking about antibodies. Yeah. I know on TWiV we talk a lot about T cells, but they're just as important as the B cells, if not more, I argue.
To summarize, if you initiate treatment at the onset of the first clinical signs, which would be 5 days, you protect against disease and you preserve pre-existing immunity. If you treat later, you improve disease outcome, but you do not subvert immune amnesia. So 7 days, treatment with drug, you have fewer clinical signs, but you still wipe out immune memory. So you have a 2-day window in ferrets to do that. All right. Now this next part is quite cool. It's unexpected. So they say, we know that naturally acquired immunity to influenza virus is better than vaccine-induced. So why is that? Can we make an animal model to try and study that? So they take ferrets and they infect them with H1N1 influenza virus. The idea was they're going to follow these, then they're going to infect them 28 days later with canine distemper virus and follow them for 5 months and challenge them with both viruses to see what happens.
And I think the expectation here is that would follow a pattern similar to the experiments that they just did, right?
Exactly.
Where they immunized them with a vaccine strain of flu, but now they're coming in with something a little more robust, infecting with the real deal.
Yeah. Right. So they infect with the influenza virus, so they get CDV first, and then they're infected with H1N1 influenza virus. They get full-blown influenza virus infection. Eventually, the clinical signs resolve. They get antibodies against the influenza virus, and then they reinfect them with CDV. And to their surprise, most of the animals die to acute hemorrhagic pneumonia with spontaneous bleeding. Major lung injury, inflammatory lesions everywhere, widespread edema in multiple lobes. They actually look at these animals using MRI. They put the little ferret in an MRI, I guess they have to anesthetize them, right, Angela?
Of course.
Otherwise, they'd be moving around too much.
That would be a very stressed ferret.
I think stressed ferret is a redundancy, isn't it?
I mean, I guess so. Yeah.
They're always stressed.
They're always energy balls. Yeah.
But the people
it would ferret a lot.
I've seen people having them on leashes as pets.
Yeah.
Yeah.
So they can track the development of pneumonia. They also take out lungs and they study the pneumonia histologically to see. And what they find that these animals are developing fulminant bacterial pneumonia when they get flu and then canine distemper virus. And these are parts of the microbiome that are normally resident in the lungs. Suddenly they're causing a problem when you have consecutive infections with flu and canine distemper problem. It's E. coli from the lung, which is expanding and causing these lung problems.
I want to go back for a minute because I think there was a misstatement here. I think you said that the animals were infected with CDV or immunized with CDV sometimes earlier, and then the flu, and then a live CDV infection. I think all these animals are, except for a couple of controls, are, if I'm not mistaken, have never been vaccinated for CDV. They're CDV naive, and then they get flu. Correct. Then they get flu, and then they get CDV.
Correct.
So they had no CDV immunity going into this. Okay, good.
That's right. Yep. Okay, so it's the expansion of this bacterial population, and they look at. So they want to know if virus is causing this. So they treat the animals with an antiviral that will inhibit influenza virus infection, and it does not. So you infect with influenza virus, then you treat with this antiviral. The animals still die of bacterial pneumonia. Okay. They even use respiratory syncytial virus instead of influenza virus, and the animals still develop this bacterial pneumonia.
I thought that was cool to show that it wasn't like pathogen specific per se.
Yeah. Yeah. So what they do is they analyze the lung tissue of these animals by RNA-seq, and they find that there are three genes encoding what are called trefoil factor peptides, TFF peptides, which are known to be involved in protection of epithelia. these are upregulated by CDV infection, and they're going to be protective against infection. But for some reason, the prior respiratory virus infection has messed with the TFFs, so that when you come in with canine distemper, that causes a big problem, and then the bacteria take over because the TFFs are not at the right levels because of the previous infection. And what the mechanism of that isn't known. At some point they say, alveolar macrophages, they're wiped out by flu infection, but then they're replaced with different ones, and maybe those are not good for canine distemper virus infection, something like that. It's something we talked about last week, right?
Yeah, this very much reminded me of last week, is that flu infection basically alters the immune status of the lung, at least by trashing the alveolar macrophages and God knows what else. Not only that, but these guys were infected with flu a month before this experiment began. So whatever it's doing is persistent for a significant period of time. Okay,
it's very interesting. Yeah.
Wow. So they get to this, I think, in the discussion. Sometimes a viral infection will lead to a bacterial pneumonia. Maybe this is part of the reason.
Yes,
some
I think
infection
that's
that
very
went on earlier.
So they say, measles is immunosuppressive. We've always assumed that other infections are because of that, but it may also be because of the effect of those other influenza on TFFs or other aspects of immunity. So, and then it turns out that if you treat early enough with the antiviral to the canine distemper virus, if you treat by 5 days, you can prevent this lethal bacterial pneumonia from happening. So it only happens when you allow canine distemper virus replication to proceed unchecked. And vaccination also prevents that as well. So you have here, I think this is cool. You have showing that an antiviral to canine distemper, which will also inhibit measles virus, will, when given by day 5 in a ferret anyway, it'll prevent immune amnesia. And it will prevent this lethal bacterial pneumonia if you happen to have had another respiratory infection in the preceding months before you got measles, right? So if you're unvaccinated, let's say you're an unvaccinated kid against measles, you get flu, and then a couple of months later you get measles, you could die. And you wouldn't know why. People say, oh, you died of severe measles, but it's because of the prior infection. And if you have an antiviral available, you could prevent that. That's the beauty of this, I think, right?
And,
we're reaching a bit because this is in ferrets, but it could be potentially like that in humans.
We are
just
reaching,
in case someone's.
But this is what you need first, right?
Of course.
A preclinical study showing there's a potential, and then you design a human study, right?
And this is a wonderful lesson in how science works, because that bacterial pneumonia thing was completely unexpected. All right?
Yeah.
But they saw that. They said, oh, what is it? The most magical words in science are not eureka, but
so that's interesting.
That's funny.
It's funny.
Oh, yeah. Or that's funny. Yeah.
So here's what they say, and this echoes what Rich said also. Bacterial superinfection after measles, like laryngitis, bronchitis, otitis media, are common. This is typically attributed to impaired adaptive immunity due to lymphocytopenia. But our data show that respiratory disease history of measles patients should be considered as a risk factor for advance to severe bacterial superinfections. So docs, put that in your questionnaire when someone comes in with severe bacterial infection after measles. Did you have flu? Did you have RSV in the last 6 months or whatever? Yeah. So this is so cool. Really amazing. It is ferrets, as Angela says, but it's a lot of food for thought. And also, we'll see this in humans, I'm sure.
I mean, it's
Good
better
stuff.
than being in mice. I want to say like that ferret models have been used for a long time for influenza and other respiratory pathogens. They're the model. So it has a little bit more clout than if it were in mice, I'd say.
I want to do 2 emails because Angela's here. Okay. and she's not going to be on for another month. So let's do this. So Angela, take Sue Ellen's email first.
Okay, wait a second. I have to find it.
Second down.
Oh, here we go. Okay. Sue Ellen writes, as the owner of several horses, I would like to take issue with Angela's rather insulting words about horses. I'd like to, but I can't. Everything she said is basically true. Aha. And what you, Vincent, said about how horses would have gone extinct if humans had not stepped in to take care of them, well, that's true too, I'm afraid. I love it when you guys get totally off track talking about non-virology things such as pigs, horses, etc. Always fun, always a good time, as Rich likes to say. Please tell Angela that I have taken the pledge and will stop eating pig products. And she's absolutely spot on, correct about all the ways in which horses are an evolutionary nightmare. Best to all, still loving the podcast, Sue Ellen. Your monthly reporter from Roswell, Georgia, where we've gone from a 20 degrees Fahrenheit on Monday to a high of 60 yesterday, headed to 70 later in the week. Well, Sue Ellen, I'm very jealous because it is not 70 here. And yes, horses evolutionarily make no sense. I stand by that.
That's a delightful email. Yeah, great
They're
email.
Beautiful animals. But yes, that was an amazing email, Sue Ellen. Thank you very much.
All right, that's Rich. Can you do the next one, Amanda?
Amanda?
Yep.
Amanda writes, hello. I recently listened to episode 1081 and that included Angela's concerns about pig farming and the welfare of pigs given their intelligence and awareness of their surroundings, describing how she witnessed the terrified look in their eyes definitely struck a chord and gives me more motivation to reduce my bacon consumption. I thought I would pass along a photo and some links that will hopefully warm your heart thanks to one pig in particular. My dog and I used to volunteer with the San Francisco SPCA in their pet therapy program, and one of our pet therapy co-volunteers was Lilu the pig. Lilu is, quote, the world's first airport therapy pig, end quote. And when she's on duty, she struts around the terminals of San Francisco Airport, bringing joy and excitement to weary and stressed out travelers. Lilu and her human have also done a lot of work to educate the public about how intelligent pigs are and advocate for their humane treatment. And she gives a link to Lilu the pig, which I previewed this email last week, and I put it on my Facebook page. Because the Lilu the pig has her own webpage, and it's delightful. And she also has a YouTube about Lilu the pig, and she provided a photo of Lilu outside Terminal 2 of the San Francisco airport. And Lilu is a hoot. I guarantee
you'll notice that her
really
hooves
great.
have painted.
Yeah.
Oh, that's
So
right.
cute.
Oh, yeah.
She's
That's
so
adorable.
cute. And
It's
her little
adorable.
friend, her little French bulldog friend with a wheelchair. Amazing.
Yeah. Great stuff. Great stuff.
So good.
Thank you so
Really
much.
Thank you.
And that dog in the background saying, God, I wish I was that smart.
Yeah.
Let me take one more, and then Dixon can take one more. So Dan writes, hello, my favorite podcasters. I've been listening to your show since before the pandemic and during the pandemic. I listen to it all the time as I was not allowed visitors, and I will continue for a long time. I live in the UK, and I would like to know how much it would cost to build a BSL 3 or 4. I know I would need a PCR, which we could buy brand new or secondhand, which is Brienne's favorite topic. We would also need 2 good virologists. They could check all the patients and staff for variants of SARS-2, as well as flu and the common cold, which would keep them busy. I have spoken to the directors, and they are interested. Okay, BSL 3 or 4 is many, many millions. Well, a 3 maybe not, but a 4, forget it. You need a standalone building. Many millions of dollars.
Right.
I would say $100 to $200 million, Rich. What do you think?
Oh man, for a BSL-4, easy. Even more. That gets up into dollar. I can probably look this up.
And then it's not only building it, even if you had the money to be able to actually manipulate those pathogens there, you need 1 million permits, and it's only specific in certain countries, and it's all regulated. And you can't just be like, okay, I'm just going to build a BSL-4 and start doing smallpox work. It's not.
Yeah, they tried that in Boston. It didn't work out.
Yeah, I think
And you'll
Boston
need-
ended up costing a lot more because of the delays and so forth.
Exactly.
Actually, I'm getting pings here that say in the hundreds, 1 to 200 million. So you're right, Vincent.
And you'd probably need more than just 2 virologists. You need a lot more
Yes.
staff than just that for either
And you
3
need
or
more
4.
than you need more than a PCR machine also.
Well,
That's
obviously that was funny. Yeah. You need
Secondhand. Yep.
Mhm.
All right. She said, I've written a poem for people who have not been vaccinated. The bell tolls. Get the vaccine into your arm. Don't worry. It will do you no harm. Don't be a selfish nitwit. Think of others that can't have it. Be wary of internet bots and trolls and be mindful for whom the bell tolls, because it tolls not for me, but instead it tolls for thee.
Well done. Thanks, Dan.
Mhm.
All right. Thank you,
Amazing.
Dan. And Dixon, can you take...
Dan's got a PS here.
Oh, he's got a PS here. Since my cardiac arrest, I have an uncanny ability to remember entire dreams. They are extremely vivid and amusing. The earlier ones are usually hallucinations. I've made a blog for you to sign up for, and he gives the address where you can read about his dreams. Thank you, Dan.
Mhm.
Dixon, can you take the next one? Linda.
Sure, Linda. Linda writes, greetings, TWiV. Enjoying your show very much. Wanted to clarify that HPV vaccine is for both girls and boys. You only mentioned girls in your discussion. Thank you for all you do. Linda Rasmussen, registered nurse.
Absolutely.
Absolutely. Yep.
Yep.
Thank you.
Definitely.
You betcha.
Charles writes, hello, Twivers. Dr. Michael Mann won a defamation lawsuit against climate change deniers, and he provides a link for that.
Good.
Too bad it was not a win against politicians. I hope it stands. Thank you, Charles.
It's an indirect win against politicians.
All right, Dixon, can you take that last one from Caroline?
Caroline White writes, if a solid organ transplant patient has an indoor cat and both of them have been tested negative for the virus, what are the chances for transmission?
So I should say that the topic of this email was T-gon toxoplasmosis.
Oh, okay. Okay.
Okay.
So that's why I was having Dixon read it. So,
Mhm.
and I think by
Gotcha.
virus she meant the parasite, right?
Right. The only virus I know of that's been transmitted on transplantation, although lots of them, I'm sure.
No,
but the
it's
one
not toxoplasma. That's what she's
I
asking.
know that, but I'm trying to.
He's expanding.
I'm
Okay.
expanding. That's right. No, I remember when West Nile virus was first circulating throughout the United States, that they had one automobile victim from Georgia that donated all their organs to whoever wanted them, and virtually everybody who got them developed West Nile, and several died from that. So that's a big problem for organ donors. Yeah.
All right. So if a solid organ transplant has an indoor cat,
Right.
and both are negative for toxo,
Right.
is there any danger to the solid organ transplant recipient?
And both are negative for the, let's say, for
toxo.
protozoan.
T-gondii, yeah.
The answer is no. They're not. What are the chances for transmission? It's
Well,
very.
Unless the cat goes out and eats a mouse, right?
That's right. Or unless they transplant the guy's brain and he's had it some other time and the antibodies have waned, which doesn't happen in toxo. You have lifetime IgG levels, and that's how
But
they
even
do
in-
epidemiological series.
Organ transplant would be what, like heart or liver, let's say, or kidney, and then is
Yeah.
toxoplasma like are they even
yeah.
latent in
everywhere.
those organs?
They are everywhere.
They are. Okay.
Oh, so you're saying they could be in the organ.
Exactly.
that's right.
That's what we're thinking.
Oh,
of,
yeah.
like the
Okay.
actual
You bet.
are they tachyzoids? I forget. Are they tachyzoids, Dixon?
Bradiozoids. If they're resting, they're called bradiozoids.
Okay, they'd be resting then. They'd be at that point.
Yep. That's why you
Yeah.
can't kill them.
Exactly. So, Vincent, what we're saying is that if there were parasites in the organ, like embedded in the tissue, that would
Okay.
then you transfer it to another person.
Right.
If you had a cat that had
Okay.
toxoplasmosis and
Or-
gave it to you, and
If the cat acquired toxo after the test, right?
Exactly.
but then the cat
Is-
would still have to give it to you, and then you'd still have to have an infection, and it would have to go to your organs, and all of that takes a while. But
Okay. So is there there is a chance that a cat can acquire toxo from a mouse, right?
Oh yeah, that's
Mhm.
probably how they get it.
If you let your cat out, that is, then a cat.
Yes. Or if you let the
Mhm.
mouse in.
Yeah, the mice can be in your home, right?
Or if you don't let the mouse in and it comes in by itself.
Well, they usually do, don't they?
Okay.
You just open the door to the mouse. Please.
That's right.
All right. There you go, Caroline.
Yeah, that's right. That's right.
All right. Thank you very much. Let's do some picks of the week.
Yep.
Dixon, you're first.
This is a rather quick look because they didn't take that many pictures, because the close-up of the moon closest to the planet Jupiter, it was a flyby. And imagine when you're flying by something at what, Rich, how fast does Juno go? Pretty damn fast, not 17,000 miles an hour. I'm sure it goes faster than that. And to get even a camera to focus on an object and to get a clear picture like they did of this moon and its surface is absolutely fabulous. The surface of this thing, it looked like somebody made it out of clay. You know, it really did. And you look at the surface of Io, and it's the most geologically active planetary body in the solar system. It's because tidal waves from Jupiter, the orbit of Io is an ellipse, and when it's far away, the planet resumes its shape. As it gets closer to Jupiter, it starts to stretch, and this creates heat. And the heat generates the molten interior of the moon, which then gives birth to these sulfur volcanoes. And it's, you would never want to visit this moon, but you would light up, maybe be 100 miles away from it and just observe the darn thing. It's by the way, it's in that movie 2001: A Space Odyssey. You can actually see that moon. They thought of putting it in there just for realism.
Dixon, the moon is a little greenish here. Is that the natural color?
I think probably that's from the sulfur compound.
Isn't that just?
Okay.
It's all
But
over
isn't
the
it just
place.
contamination, like pollution? We see it as green as opposed to it actually being like it.
What's it been polluted by?
No, I mean our atmosphere. Like in our atmosphere,
No. This is Juno.
Juno.
This is a satellite that we sent to Jupiter, and it took the picture.
Oh, never mind. I thought Vincent was saying also our moon, and I was like, no, it doesn't. I thought he was referring to our moon
Our moon
as well.
is made out of green
and
cheese.
I was like, if it looks green, it's probably just pollution. Never mind.
Angela, I wanted to know if it was algae.
The moon is moving so slow; it has algae.
That's right. Uh-huh.
Yeah. Well, that's cool.
Yeah.
Very nice. Thank you, Dixon.
You're welcome.
Angela, what do you have for us?
So I have a pick on whales, because if you're a TWiV listener and if you listen to at least like 3 or 4 episodes with me, you're very aware that marine mammals and whales are like my favorite thing, and the ocean. So I found this website from these 2 scientists, these 2 early career scientists. So it's whalescientist.com, very easy to find. And there are 2 scientists that run this website. One of them is Dr. Anais Ramili, and she's a postdoc at McGill. And I didn't actually know about this, so I have to go find her now. I'm going to go find where she is on campus and go talk to her. And then the other one is Naomi Matthew, and she's a PhD student at the University of Louisiana at Lafayette. And basically this website has a whole bunch of it's all about awareness of marine mammals, not only whales specifically, also like manatees and seals and other marine mammals. And they have little clips, little blog posts about, one of them is toxic chemicals from plastics found in marine mammals. Other ones are like interesting facts about like, why do baby whales and dolphins have whiskers? Like just a lot of like very fun. Well, they're not all fun necessarily. Some of them are fun and some of them are more serious, like talking about contamination and pollution, plastics in the ocean, how that affects different marine mammals. They also have a section about whale scientist stories. So they interview other scientists, a lot of them graduate students or trainees, and just talk about their research and where they are, how they're, what type of research they do in whales. And I thought it was just really, really amazing, this webpage, whalescientist.com. It's so cute.
Very cool.
And whales are like the coolest.
Angela, they have one section. Why should early career scientists engage in science communication?
Yeah.
It's good.
It's amazing. And
they don't have any sea otters here. Do sea otters count as marine mammals? I would think they would.
Well, so it depends. When we were in vet school, they included them in marine mammals, but I remember there was this whole like controversy about it because they're in freshwater. But then also some are like the dolphins, the river
I'm
dolphins
talking
are
about sea
freshwater.
otters. I'm talking about sea otters.
Sea otters.
Well, in the sea, yes. So sea otters, if they're in the sea, then they are. Yes,
Okay.
they are marine mammals as well. So maybe
But
they
they-
just don't have any on their page
They don't
yet.
spend enough time submerged to qualify. There's got to be a fraction of time submerged to qualify you as
Polar bears are officially marine mammals.
Is that right?
And yeah,
Huh.
yeah.
Okay.
You know, the polar bears actually have webbed, their interdigital space is webbed for
Cool.
swimming. They also
That's all
have
going
like
to change,
this,
though. You know that's going to change, right?
I know, but
Yeah, just like
Unfortunately.
Pluto
They're now-
getting kicked out of the solar system.
They're
Exactly.
They're now hybridizing with grizzly bears,
Oh, wow.
which is an interesting hybrid to think about. A grizzly bear that can chase you into the water and continue to chase you.
or think about how beautiful they are, not only the aggressive, terrible things. I mean,
Oh,
we
they're
always
gorgeous.
think of them
They're
as so
gorgeous
negative.
animals.
They're so beautiful. Um,
So,
Yeah.
Angela, stay tuned. I almost had a marine mammal pick lined up for today, but it got kicked off because of other things. But I'll keep it in the queue for later on. You'll like
Okay.
it.
So last week's Nova show, I think, was all about the evolution of whales.
oh,
Where
really?
do they come
That's
from?
right.
Yeah.
Yep.
Where do they come from? And they come from land animals, of course.
Well, yes, because we went from Earth
That's
back to
right.
the ocean, which is why when
That's
you
right.
see dolphins, like their fins, their pectoral fins, they actually have phalanges, which a lot of people don't know.
Yep.
They have like little fingers. It's so cute. And some
They
of them
do.
even have vestigial hip bones still, like just embedded in their muscle tissue. They don't actually have legs, but they have like these two little vestigial bones, these little
Yeah, yeah.
like
Hmm.
that don't.
They
do anything.
saw humans coming and said, "I'm out of here. I'm
It says,
going back
forget
to
it.
the sea."
Forget about it.
Rich, what do you have for us?
Okay, I have what I'm calling a bundled pick. It's got 4 links in it. It's not as bad as it looks. So I'll make it easy. Okay. People who've been around for a while know that I volunteer for this organization called the Brighton Collaboration, which is an organization dedicated to developing international guidelines for vaccine safety. I think that their most notable accomplishment so far has been in developing case descriptions for various adverse effects in vaccines that are published internationally that physicians can use to try and understand what they're dealing with when they see a bunch of different symptoms like Guillain-Barré and that kind of stuff. So recently, in the subgroup that I work with deals with trying to develop uniform templates that would describe various vaccines that people can refer to understand what the vaccine consists of and a little bit about adverse effects. And in the course of this development, we became aware of the fact that benefit risk is a real discipline nowadays. There is actually a whole discipline that's about benefit risk analysis. And we got a couple of these people who are experts in this to work with us and basically spin off from the templates another bunch of templates that are up and coming that are a benefit risk analysis of vaccines. This is something we talk about all the time, is to be able to actually measure the value of a vaccine in terms of what are the benefits relative to the risks. And this is an attempt to actually quantify that in a real reproducible way. So this group, a subset of the committee I work with, recently published a commentary in Vaccine along with their first digitized template of the benefit risk assessment. And so the main link here is called Benefit Risk Assessment of Vaccines. That's all you really need, okay, to describe this process, and that'll link to the other bits. The other link I have here is the actual article on the benefit risk assessment tool that they have developed. That's the main part of the pick. All of this reminded me while I was going through this. The guy whose brainchild the Brighton Collaboration is. This next bit is a repick. The guy whose brainchild the Brighton Collaboration is a guy named Bob Chen, okay, who thought up the Brighton collaboration. He is a vaccine safety expert. And he also chairs this subcommittee that I work on. So I've gotten to know him reasonably well. In my mind, he is notable for a variety of things, but among other things, remember the old Wakefield paper that set the planet on its ear? He and another guy named DeStefano wrote a commentary that appeared in that issue titled Vaccine Adverse Effects, Causal or Coincidental. All right? And that commentary debunked Wakefield's paper in the same issue that Wakefield's paper was published. And if you look at it now, it debunked it in all of the ways that absolutely made sense. It saw all the flaws, it laid them out, and it said, this is nonsense. And it was completely ignored. Okay?
Wow.
So I just
that.
want to acknowledge Bob and acknowledge that fact and how stupid we really are by putting that link in here too. Lastly,
I didn't know that.
I picked that a long time ago, I think. I hope. Actually, at the time, I don't think it was open access, but now it is open access. You get it through, what is it, Science Direct, okay? And that's what this link will do. And it's a fascinating paper to see it debunk everything. While I was doing all this today, I was multitasking because the group that I sing with is hawking singing Valentines on Valentine's Day. We have several quartets that are going to go out. You can order a singing Valentine, and we'll send a quartet to wherever your loved one is, and we'll sing 2 love songs and give them a rose and a card. So I've got a link in here. So Austin locals can order a singing Valentine from us. Okay?
I love it.
And if you're not in Austin, and you want a singing Valentine, go ahead and Google singing Valentines and even singing Valentines Barbershop Harmony Society or something like that. And if you're in a reasonably populated area in the United States or even elsewhere in the world, you will find a local chapter through whom you can order a singing Valentine. So I highly recommend it. It's a hoot.
Cool.
Done.
Thank you, Rich.
You're welcome.
When this episode drops, February 11th, that's Sunday, it will be the International Day of Women and Girls in Science.
Yay!
And I have for you a United Nations page all about that. And, it has facts about women in science and has some links that you can look at to tell stories of women in science, different areas of science, and why we are doing this, and so forth. So
Excellent.
This was started in March 2011.
Cool.
And it explains why we mark international days like this. So check it out. It is perfect timing for TWiV drop.
Vincent, can I give a shout out to someone that came to visit me at McGill this week? So
of course,
I was just sitting in my student room waiting for my centrifuge, taking some sips of coffee, as you do. Life goes by while waiting for the centrifuge. And one of the other students on my floor, Dan, he's doing his master's at the Vidal lab. He just shows up at the door and he's like, "Oh, Angela, my friend came from Calgary. She's visiting me in Montreal." And he said that she heard my voice down the hall, looked at him and said, "Is that Angela from TWiV?" And he's like,
that's great.
He's like, "Oh yeah, she's on this floor." And she had, he had told her a week before, but at that moment she heard me and then came over. So her name is Simone. She's from the University of Calgary here in Canada. And she's a PhD student. And I wanted to just give her a shout out. She was so nice. She came and spoke. She's a huge fan. She's been a fan for years. And we talked for 20 minutes. And it was just really nice to have her come by and say hi.
Love it.
Yeah.
That's nice. Yeah, we had a visitor this week at the incubator from Chile.
Nice.
Pablo was visiting New York City and came by.
Cool.
And he's a listener also, but he works as a translator.
Oh, cool.
Translates between Spanish and English. So I said, "How would you like to translate my virology lectures into Spanish?" And he said, "Yeah." So he's going to do it.
Oh,
I
wow.
Outstanding.
I think that could reach a lot more people.
Yeah.
Oh, definitely. Yeah.
Half
And
the Earth's population, I think, something like that.
Is that right? And so we're going to do that. And then on the live stream Wednesday night, one of our listeners, Veronika, is from Russia, and she's going to do it in Russian.
Cool.
Excellent.
Those are two big ones. I want Chinese also. So if anyone out there wants to do Chinese, I don't know what other languages. Is
French.
Portuguese a big one?
French. Portuguese is big in Brazil, of course, because there's
Brazil and
a
Portugal.
lot of medical schools in Brazil. But French, I think you should pick next, because half of Africa speaks French,
That's
as
right.
well as France. And Belgium and some others.
Here in Quebec.
Even in Quebec.
Quebec. What about Haiti, right? Don't they speak French?
Yeah, they do. Yeah, they do.
Also.
They do.
Yeah.
They do.
All right. International Virology. That is TWiV 1087. You can find the show notes at microbe.tv/TWiV if you have questions or comments. TWiV at microbe.tv. If you enjoy these programs, please support us. Microbe.tv/TWiV. No. Microbe.tv/contribute. I don't know why I said TWiV. I've got TWiV on the mind. Dixon Despommier, trichinella.org, thelivingriver.org. Thank you, Dixon.
I promise I won't forget this episode.
Rich Conda is an emeritus professor, University of Florida, Gainesville. He is currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. This is a great episode. Good time.
Angela Mingarelli is at McGill University in Montreal. Thank you, Angela.
Yeah, this was so much fun today. I can't wait for the next TWiV already.
Well, we try and put animal-related things when you're on, you know.
I appreciate
Exactly.
that.
Exactly right.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.